invitation to.
All right. Well, thanks, Steve, for coming on our show. And we're going live again. And Steve has been such a generous patron of this community for youth and families. So really appreciate you coming on again in spite of all the wonderful things you're doing out there in the world. So Steve, I don't want to start off with your CV, as a lot of people know a lot about you. So maybe you can just briefly tell us what are you doing at University of British Columbia, and then we can get started.
Thank you. It's great to be with you again. For those of you that don't know me, I'm a professor in the Division of Neurology in the Department of Medicine at the University of British Columbia, where I've been on campus for in faculty for about 35 years coming up. And I'm also run a biotech company, Kinexus Bioinformatics Corporation in Vancouver. And I'm going to present a little bit of the work that we're doing on a clinical study that we've been performing for, I guess, 2.5 years now. And I'm also the vice president and the co-chair of the Scientific and Medical Advisory Committee of the Canadian COVID Care Alliance. So, but today I'm going to really more talk about what it is that we do in our company. But because that I'm on faculty at UBC, and I am going to be talking about some things that relate to my university research as well, I have to give a conflict of interest disclosure because I'm, again, the president of Kinexus Bioinformatics, and I do have about 75% ownership in the company. And I'm going to be talking about some of what we do in the company. So just be forewarned. Okay, and also the views that I'll be expressing are my views. They don't represent necessarily the University of British Columbia or the Canadian COVID Care Alliance, although I think most of us in the Alliance are all thinking pretty much the same way on the Scientific and Medical Advisory Committee. So we've endured this COVID-19 pandemic now for over 2.5 years. I think the actual pandemic started in November, actually, of 2019. Officially, it's thought that the first wave commenced more in mid-March. And the idea was that it actually somehow didn't come from China, but it came via Europe to come to Vancouver area. And that's based on sequencing studies that were done of the virus. But those sequencing studies did not commence for the genomes of these viruses until really more April, May. And so we may very well have had an earlier variant that came and went and got displaced, as I'll show you shortly, often happens. But we have at least 7 waves now that have been distinguished. And these 7 waves, the first one was thought to be very small, but that really is a reflection of the inadequacy of the testing that was done at the time. And however, we did have a death peak that's relative to the size of the incidence peak here. The first peak here that's showing is.
Do you want to see this? Because I took the camera off.
On the screen that we're seeing is actually the incidence, and then the bottom is deaths. And so we can see we have a big death peak relative to the size of the incidence. And so later we got another big peak with the Omicron towards the end of December of last year and the beginning of this year. But however, when you look at the total number of people infected versus the number of deaths, it actually turns out that it's about 4 to 5 times less lethal than the Delta strain that preceded it. So when we look at really the way that the virus and the pandemic has transpired, we actually see that people that are very young are at extremely low risk. I show you here that they represent under 20-year-olds less than 0.1% of all the deaths from COVID-19. It's really the elderly the greatest risk. And they account for over about 82% of the deaths from COVID-19. Now, that's with COVID-19. We actually have to think about whether they actually died from COVID-19. And the statistics that seem to be most consistent is that about half the deaths of people with COVID-19 are due to COVID-19 as opposed to other morbidities that cause their deaths. Okay, so as we know, this virus did arrive from China, at least initially. Some people say, like December 2019, there was reports of this disease in the Chinese community. It's thought to have been from the Wuhan area. I think most scientists that really study this now think it was actually a lab leak, because there is genetic editing of the virus that has been evident when you actually look at the structure of the virus at its genome sequence. But it's very related in its structure to the SARS-CoV-1 virus that hit us in 2003 mainly, and also the MERS virus, which is affected people in Saudi Arabia. Interestingly, the SARS-CoV-1 disappeared within the year without any vaccines, without any treatments. It just apparently disappeared. And MERS, pretty much the same thing. I would think that actually the virus didn't disappear. The community developed antibodies as it mutated to a much more benign form. It didn't cause the degree of illness that it first had. And that's sort of what we've seen happen with SARS-CoV-2. And there are at least examples of previous viruses, cold coronaviruses, at least 4 of them, that are very common. Again, they cause the cold. They make you ill for a short period, but they're generally not life-threatening or very severe. So the virus itself has about 28 proteins that really decorate the surface of this spherical virus. So these spike proteins are complexes, actually, of 3 spike proteins per complex. And they really stick out, and they form that crown-like appearance that gives the name coronavirus. And with the sequencing efforts that have been done worldwide, it's evident that there's probably at least 15 to 20,000 variants now that have been identified as mutated forms. There's about 30,000 nucleotides in the virus, so any one of those could give you a entirely different mutant, and certainly different combinations of those mutations give you different mutants. But what's really important is that very few of those mutant forms actually compete successfully. And there is, in fact, variants of concern where these seem to be very infectious, and they replace all the existing earlier variants, as we'll see shortly. So this is the virus, and it's many proteins, but only 3 of them on the surface of the virus: the spike, the envelope, and the membrane protein. Now, when you have natural immunity and you're infected with a virus that comes in through your airways, and you have an antibody response that's actually against all of the proteins in the virus, the antibodies that you make that are against the surface proteins of the virus are going to be the most useful to you. And we know from other viral infections that if you develop immunity, it usually lasts for at least 2 years or more. And this is because we have the establishment of those cells that produce these antibodies, the B cells, these particular lymphocytes of your immune system. They can become what we call memory B cells or plasma B cells. And these can in fact last for decades. And there's a lot of evidence to support that. So people even today can be measured to have antibodies still in their system that recognize the original SARS-CoV-1 virus, because those B cells that originally produced it are still in their blood system. So one of the things that's important to understand is that when you have a respiratory virus that infects you, you make IgA and IgM class antibodies. These are antibodies that are secreted into the airway spaces, and they encounter the virus as it first comes into your body. So they provide the best protection. And the virus manages to get inside cells by binding to this protein called ACE2, which regulates blood pressure. And by latching on to the surface of a cell through this receptor, once attached, it's able to then initiate a program to enter inside the cell and then replicate and produce more of the virus, which includes many of the other proteins that are in the virus. Okay, so we started at Kinexus with a clinical study to really look at what kind of antibodies is it that people are producing against this virus. We thought that if we could identify the parts of those 28 proteins that elicit a strong antibody response, then we might be able to go ahead and take pieces of that virus and inject that into people, and they would make antibodies that in fact might be protective for those individuals. Our company has been making antibodies in rabbits for about 12 years. We made about 1,600 antibodies by just taking short parts of proteins that we're interested in making antibodies against and injecting them under the skin of the rabbit, and then they have an immune response, just as if they're being infected by a virus or some other pathogen. So we started our study in March of 2020. We have the capability in our company, by the way, of making all of the proteins of the virus in individual pieces by chemical synthesis artificially, and we can put them onto membranes that we can as individual spots for the individual parts, and then take blood from people that have antibodies. For example, they've recovered from SARS-CoV-2, and then look to see if those peptides, those parts of the virus are sticky and capture the antibodies. And then we can tell if an antibody has a recognition of a particular part of the virus. So that's sort of like how it works. And we started recruiting people in our study, a lot of by word of mouth, actually. And we had dentists that approached us early on because people involved, dentists were, working around the mouths of people, and many of them actually did get sick, although it seems some of them got it from a dentist convention here in Vancouver that spread even beyond that, but they all came down with these symptoms. And so they were early in our study, and we were able to use people who had COVID-19 to look first what part of these patients who had recovered, so therefore their immune systems were very successful in taking out the virus, which parts of the virus did they make antibodies against? And ultimately we recruited about 3,500 people up to really, I guess, November of 2021. We had even babies that were blood samples from that were 6 months old to 90-year-olds that we were able to test. And there are roughly slightly more females than males, but pretty similar. And I would say about half of those people, a little under half, had symptoms of COVID-19. Many of them were confirmed by PCR to have COVID-19, but you'll see in a moment why we didn't have more confirmation of PCR results for these participants. Now, when we look at those participants that agreed to come into our study, and we look to see, well, when did they actually get COVID-19? And this result was very interesting because basically 3/4 of all the people that had COVID-like 19 symptoms were from November, December of 2019 and January, February, March of 2021, 3/4 of all the people in our study. So I showed you earlier from the incidence of reported cases that this was a tiny peak. But when we look at our study for people who said they've been sick, it's the bulk of the people in our study. And in fact, those people we've tested, and they have antibodies against the virus. And we tested them soon after they actually had these symptoms. So it's very likely that they actually were making antibodies against SARS-CoV-2, as opposed to maybe cross-reactivity with earlier versions of coronaviruses that have some similarity. So that tells me that, and makes more sense, that being in the Vancouver area and a gateway to China, and a high degree of traffic back and forth between the Vancouver area and China. We have a lot of very successful Chinese business people that go back and forth that live here. That makes more sense that we were in fact hit by the virus early. And it also means that many people already had antibodies against the virus early on in the pandemic in the Vancouver area. And as we tested more people outside the Vancouver area, especially in the Ontario area, we find the same thing. Most of the people in Ontario that we test actually had it in the first wave, the symptoms of COVID-19, which we confirmed. Okay. So what about that test? Well, we originally tested all 28 proteins by slicing them into overlapping pieces of over 5,000 or 6,000 different fragments from these 28 proteins, all arrayed out. Here you can see in the slide an example of an array that has about 300, this is 110 membrane, but we made membranes that had as many as 900 spots at a time, different parts. And we took many of these people early on that had COVID-19 to try to figure out which parts did they make antibodies against. We found certain parts were very immunogenic. That means that people make antibodies more easily against these particular parts. And then from those parts that were common amongst our people, our participants, we then made these membranes. Now, what you're seeing here is basically in the first column of these different blots, these are called immunoblots. If a person had an antibody against a particular part, then it shows up as a very strong spot. The bigger and darker the spot, the more antibody that person has against that particular part of the virus. So here we see an example of about 9 people who are all different people. They're all having an antibody response. They all recovered from COVID-19. And when you look at the patterns, they're all different. Here you can see one spot that's consistent. That's a control spot that we have at this position to make sure that these screens and our assay ingredients are working properly. So the pattern, while there's some similarity from one person to another in certain targets from the virus, everybody has a different pattern. And again, this is only looking at about 110 of the markers that we picked, of which there were several hundred that we identified. Okay, now what's interesting is that when you take the same people and you look at their antibody response 10 months later, the pattern is almost the same for each individual. So while they differ from individual to individual for the patterns, for the same individual, which is shown in the rightmost columns now, for what's in the left column horizontally, it's the same person. They have the same pattern and it's the same intensity. And this is important because normally when you develop an antibody response, you get in a matter of a week or two a nice response. It stays high, and then as your immune system takes out the pathogen, the virus, bacteria, or fungi, then your antibody levels will slowly decline over a couple of months. But here you can see it's maintained, and I think that's because we're in a pandemic and we're constantly being exposed to the SARS-CoV-2 virus, or at least the next variant of it, which again boosts our immune system, and we have an antibody response. And so we can see people even 2.5 years later from when they're the only time they remember of being infected, having these symptoms, still having detectable antibody levels from natural immunity. Now, we took that 110 membrane test and we narrowed it down to a 41 marker test with a control. This is shown in the bottom right corner. That's to make sure that the screen is working. And here you can see a few more different people. And again, the patterns of the immune responses differ, but these were the most common responses we saw in the first data set that we did of 500 people. And then the other 3,000 people, we used this particular screen. And you can see here that the top spots correspond to the spike protein parts, including the receptor binding domain. This is the region of the spike protein that binds to ACE2 and allows it to get inside cells. And so the idea is if you have an antibody that binds to that receptor binding domain region, it may block the ability of the virus to enter inside of cells. So there's another 9 other proteins that are tracked as well. Now, with these screens that we've done, one of the things that was interesting is, obviously, if you're vaccinated, the RNA or the adenovirus DNA that's being delivered inside your cells is getting those cells to make the spike protein. So you should get an antibody response for the top half of our screen here, the 41 marker screen. The bottom proteins, you don't expect to get a response to the vaccine. You might have some cross-reactivities weakly with other coronaviruses that you may have been infected in your lifetime, but you shouldn't be getting spots at the bottom if you've never been infected with the virus before, but you get vaccinated, you should see spike responses, okay? One of the proteins that is tracked here is the nucleocapsid protein. Now here, spot D1, D2, D3, and D4, shown in the map here, are where this person has a very nice nucleocapsid response. This person also picks up at least a nucleocapsid response. This person has no nucleocapsid response, and this person has a very weak nucleocapsid response. So the reason why this is important is that in all the studies that are done to date, where people are looking to see, well, did you get your immunity naturally from an infection with the virus, or was it due to your vaccination? In the case of the vaccines, it's the spike protein that you're measuring, but you could have also gotten the spike protein from a natural infection. So they look at the nucleocapsid protein levels, and if they see nucleocapsid protein, then they can make an assumption that you've been infected with the virus before. But as I've shown you here, in fact, you can have people that have a good immune response, but it does include antibodies against the nucleocapsid protein. So automatically that means that there's dramatic underestimates of the number of people in Canada who've actually been infected, because when they have measured it, they've exclusively looked at the nucleocapsid protein response, whereas we're looking at about 10 different proteins. Okay. So this occurrence, though, of a widespread immunity, it's not just observed by Kinexus. And in fact, I-Corps Blood Services early on contacted me, and they noted by September of 2021 that over half the people that were unvaccinated that they tested clearly had antibodies against the spike protein. And they did a study in Lacrete, Alberta. This is a northern western part of Alberta, so it's very rural. So they found that 89% of the people that were unvaccinated that they tested had antibodies against the spike protein. And so if you see this in a rural area, you can only imagine that in a urban center where you have high density, that clearly the vast majority of the people must have already been infected and had natural immunity back even a year ago. Okay. Now, since then, of course, Omicron hit, and even the Canadian Blood Services, using the nucleocapsid protein as a marker for natural immunity, concluded that nearly 40% of all Canadians had been infected with Omicron. So again, that number is probably double, because about half the people we test don't have antibodies against the nucleocapsid protein. And other studies that were done in the States showed a seroprevalence now in children, about 75% of the children. Even in England, they found just really at the beginning of the year that 97% of their secondary school kids actually had natural immunity against the virus. And about 62% of the elementary school kids. Bonnie Henry here in BC is the co-author on a preprint that just came out, and they conclude that around 70% of all of the students that are tested in BC have natural immunity already. That doesn't seem to have affected her decision-making on the vaccination requirements for healthcare workers, but it does sort of indicate, because she's a co-author on the paper, that she's certainly well aware that the virus has actually spread throughout BC, and most people actually have natural immunity. Okay, so we started a second study in June of this year, and the reason why we did this was that we have these new variants of SARS-CoV-2 that have emerged. We found in our study many people that had been infected in the first wave did get Omicron. Not all of them, and many of them that did, it was very mild. But a large number of them did actually get infected again, even though they had natural immunity with the Omicron variant. And so the question really was, could our test monitor the presence of the Omicron virus and its variants? And how long was our natural immunity actually continuing? And the big question really, though, was how good is the immune response to the vaccines? Because what we're seeing from the epidemiology studies is that after your second shot of a vaccine, it seems as though within 6 months, you have really lost your immune protection. You don't have detectable antibodies, according to these other studies. And you also end up getting sick with COVID-19. In fact, what we're seeing is people who are vaccinated now, especially with booster vaccines, seem to be more likely to actually get COVID-19 in the future after that period of protection has subsided. So you're actually, soon after you're vaccinated, you're at a slightly higher risk of getting COVID-19. We've known this since the first shot from data from Alberta, where we look at breakthrough cases. It all happens within the first, the initial breakthrough cases is in the first about 9 days. There's a very high peak right after you're vaccinated. The risk, like you would think that you have a high risk and then you get vaccinated, and it'll take, maybe a week or two to get your antibody levels, and then your risk will go down. But instead, when you look at the data, the risk is relatively low, and then it goes up in the first 7 days, stays high, and then comes down as you start making antibodies. So initially, when you're vaccinated, you're actually at a greater risk of getting COVID-19, especially when there's a pandemic and you're in a wave. Then you have protection, and then the immunity wanes. So we wanted to actually study this more. Now, so it's still early in our study, but I did do some analysis, and I would say that at this point, and I'll show you an example at the end, it looks like your natural immunity actually is reduced about 10 to 15%, I would say, based on quantitative analysis against the spike protein if you're vaccinated compared to the natural immunity. So again, one of the other things I wanted to know was, is there a relationship between the natural immunity and vaccine vaccination of people. We know from published studies, for example, that if a person has not been vaccinated before and they look at nucleocapsid antibody levels, they're very, very high in people who've been vaccinated, even though, as I pointed out earlier, in many tests, they don't have the sensitivity to pick up that nucleocapsid detection. Now, when you have someone who's been vaccinated and then they get COVID-19, the nucleocapsid response is much lower in terms of making antibodies by more than half of what they saw in that study for people who were unvaccinated that got infected. So it appears as though you're blunting your immune response if you've been previously vaccinated and then you get COVID. You're not getting as strong or robust a natural immune response. Okay. So for those of you that are interested in enjoying our study, I have a URL that's posted here. Maybe Sarah can take that URL and put it with the video for you in the future. We do have to charge to do the analysis. We have no grant funding to do this. So our participants are helping us fund our study. And we hope to get all of our results published very shortly. But we are being very careful, and we're doing over a long period of time so that we can make some real definitive statements that are statistically backed. Okay, so as I mentioned, you have all these variants of SARS-CoV-2 that emerge, and you can see here in British Columbia, these colors correspond to the different variants that were predominating over time since the beginning of the pandemic. And you know, the Wuhan strain was the first, but even the Wuhan strain has variants, and the variant that came from Europe replaced the Wuhan strain here in British Columbia. And we had alpha and then beta, and then like delta was a big wave that we got. These are shown in different colors, like delta wave here is in blue. Then we started getting the Omicron wave. And to illustrate just how fast this competition between an older variant that is already quite infectious, that predominated and beat out all the other variants to infect people, with the Omicron in December, it basically within a month displaced the Delta variant as the predominant variant here in BC. And this phenomenon has occurred throughout the world. And you can see here in the red with the Omicron that variants of the Omicron started to emerge. And of course, now I don't have the latest data, but the B4, the BA.4 and the BA.5 variants are the predominant variants now in Canada, and they've completely replaced the original BA.1 and BA.2 variants, which is interesting because the vaccines that are being made now, the so-called bivalent vaccines, which actually are not bivalent, they actually have the original testing is on the combination of the Wuhan spike protein with the BA.1 spike protein in the tests, but based on further tests with the BA.4 and the BA.5 mixed, studies done in like 8 mice, no human studies, they went ahead and approved this for the vaccines that people are injecting into themselves now. And so much of the spike protein that's in there is actually corresponds to the original variant, which is long since gone. It's pretty much extinct now, except in probably freezers and labs. Okay, so when you look at these mutations that occur in these variants, and I know this is a very busy slide, but it's the point I want to make becomes quite apparent. Each column here is a different variant, and these first 3 columns is the Omicron variant. And then we have the other variants, alpha, beta, gamma, delta. So each column is a different variant. And the rows correspond to a particular mutation that's occurring in the spike protein. So if there's a mutation, it's either showing up in these variants as either green or yellow, okay? So you can see, compared to the other variants, we have a lot more mutations in the Omicron variants. In fact, there's over 75 mutations in the overall virus, but about 34 of them are in the spike protein alone. And this seems to confer an extra advantage for the Omicron variant to bind better to ACE2, and maybe some of the other mutations also allow it to evade the immune system better. And so it's more infectious than earlier variants. Now, the real question is, if you have all this change in your structure because of these mutations, well, will the antibodies that you made against the original Wuhan strain or other variants work to recognize the new variants? And the answer is absolutely, because you have about 1,273 to 1,278 amino acids, and these mutations are showing you where you have in at least one of the variants a mutation occurring. So what you see is that in these mutations, you're getting only about 2.7%, even with the Omicron variant. Most of them is less than 1% change in the overall structure of the spike protein. You can't change it too much with these mutations because then it won't work. So the chances of having a mutation that actually improves the binding is relatively low compared to having a mutation that actually reduces the binding and is a disadvantage to the virus compared to the earlier strain from which it mutated. So the bottom line here is 97% of your antibody response is also going to be intact, because 97% of the structure of the protein is the same. The real question is, well, are those parts where we identified people making antibodies against that were very immunogenic, are those the parts where people actually have the mutations in these variants? And so here is an example of one of the larger immunoblots that we did early on. Looking at the top part, spike protein, and here we have the nucleocapsid protein, and here we have the membrane protein. Again, the membrane protein and the spike protein are on the surface of the virus. So any antibodies against these are going to be particularly useful because they'll tag the virus so that the rest of the immune system can recognize it and take it out. Now, these spots that you're seeing here, this is an overlay of about 9 people that had recovered from SARS-CoV-2. What I'm showing you with the yellow bands are the regions where the mutations occur in the Omicron version. And so with few exceptions, about 90% of the regions where the mutations are occurring are not very much immunogenic. They're not the regions that we make antibodies against. So we expect that actually we have a very good immune response even to Omicron. And that explains why people that are vaccinated with the original Wuhan strain still have some protection against even the Omicron strains, because those antibody responses aren't going to be in the regions where people make antibodies against. Those mutations aren't in those immunogenic regions. So I think that's good news. It also means that if more than 40% of our population has been infected with the Omicron strains, and it's probably more like 80%, then in fact, our antibody response is even better for the most recent versions, because we've not only boosted what we already have, but we've also found other antibody producing cells that we now stimulated to make antibodies as well. There is this what we call original antigenic sin, which means that your initial immune response tends to predominate, but you may still make other antibodies as well. Okay, so we got these 6 vaccines now. 4 of them were approved, last year. 2 of them came on the market this year. The 2 new ones, the Novavax and the Medicago, are not genetic vaccines. They don't deliver the information to make the spike protein in your own cells. They actually are lipid nanoparticles that are derived from either plant insect cells or plant cells that have been ground up. So the surface of these actually express the spike protein on their surface. They've been genetically engineered, these cells, so that they already have the spike protein on them. They're ground up into little lipid nanoparticles, and then they're injected into your skin. So your immune response is directly against these, we call them exosomes, that have the spike protein on them. Whereas the traditional, well, it's become traditional now, COVID-19 vaccines are the ones which are actually genetic, and you're producing the spike protein on the surface of your own cells to elicit the immune response. Now, what's important here is that these have been described early on, especially the genetic vaccines, as being like 95%, effective. And that's relative risk reduction. And it's important to understand that what that means is if you have a population and you see in those 2 populations a vaccinated and unvaccinated population, maybe out of 20,000 people in each group, 160 that are unvaccinated get it, and around 10 in the vaccinated group get the COVID. And then you compare, well, it's 10 compared to 160. And then you look at that and you say, well, it's something like around a 90% or so efficacy. But you have to remember, originally, most people in the study didn't get it at all during that period of time. That's 6 months. They didn't encounter it. They didn't get infected in both groups. The vast majority did not. So we look at then, well, what's the percentage that if you were vaccinated compared to unvaccinated that you would have gotten it? And so for all these vaccines, it actually works out to around a percent difference. So that's called absolute risk reduction. Now, in the case of the Medicago, you can see that 70%, 71% risk reduction. So it wasn't even very good from a relative standpoint. The Novavax was a little bit better. Now, again, this absolute risk reduction is really important to figure out the protection that you're getting from the vaccine. And again, with the vaccine, you're only making antibodies against the spike protein. So there's an issue there, and you're not establishing immune memory. And because you're injecting it into your muscles in your arm, the kind of antibodies you produce are the IgG class antibodies. Primarily, they're very good antibodies. They last about 21 days, whereas IgM and IgA antibodies is around 5 to 6 days. The IgG antibodies are in your circulation. They travel all around your body, but the concentrations of those particular antibodies, the IgG antibodies, in your lungs and airway spaces is very low. So as a consequence, people who are vaccinated, they don't have a very high level of these particular antibodies at the site where the virus is coming in to infect you. So the virus gets in, replicates, and leaves. So that's why people who are vaccinated can still get COVID-19, and they can still transmit it to other people because of the nature of the antibodies they produce. And they don't keep producing it. They don't establish these immune memory, and it's only against the one protein rather than against the whole virus. So that's why the natural immune response is so much better than the COVID-19 response. Okay, so as I mentioned, everybody accepts now that even though you're vaccinated, you can still get COVID-19 and you can still transmit it with viral loads that are pretty much the same between unvaccinated people who get COVID-19 and vaccinated people. There's an argument that, well, yes, but you're not going to die from it as easily. You're not going to go to hospital as easily. However, there's no data, no clinical study that actually supports this assumption. Bear in mind that more recently we have a more benign virus, so you're less likely, if you're going to get infected, to get that sick anyways. Secondly, there's a high degree of natural immunity in the population. So even if you vaccinated people, almost every person that we've tested that's been vaccinated has natural immunity. So how can you tell? There's just no data to really support this. So there is evidence for a temporary reduction in ICU cases and deaths, but now what we're seeing is if you're 3 times more likely, for example, to get infected if you've been vaccinated, this is what the data showed out of the UK last Christmas, if you're 3 times more likely to actually get COVID than an unvaccinated person. So if you're more likely to get it, then you're also more likely to probably have a severe reaction to it in terms of the COVID if you're infected. So it's hard to see that. And now we're seeing that after you get your temporary reduction, there's higher cases. There's more people that end up going to hospital and dying. So you have more people that are dying that are triple vaccinated than are double vaccinated when you adjust per capita. So this is the problem. And that's why, in Israel, they went to the 4th shot, and even some people are on their 5th shot. I won't be surprised if in 3 to 4 months, we're going to have people doing their 6th shot. Now, people have argued, well, this is normal that, we give vaccines. Sometimes we need 3 boosters, but you have to remember that in those studies where they're doing this like 3 boosters or 4 boosters, these are babies. These are children under 2 years of age that their immune systems don't work as well as adaptive immune systems. They're very good, robust, innate immune systems that are highly protective, but to develop that antibody response, that adaptive immune response, that's when they're doing these extra boosters. We don't give these kind of number of boosters to adults normally, 3 shots, 4 shots. Okay, so you might make a case for people that are very high risk, very elderly, that they, with comorbidities, that the injury from the vaccines and the risks of that might outweigh, or be outweighed by the risks of actually getting COVID-19. But I think that's a very narrow window. And it's an entirely different presentation to talk then about the vaccine injuries, of which it's remarkable just how much injury there is associated with these vaccines. But suffice to say that that's possible. But what I'm really interested in, this is the last slide, is do we reduce our immunity, our natural immunity, if we go and get vaccinated afterwards? And I'll give you a case study. Here's an individual, male. He'd be in his 40s, and he was vaccinated on June the 16th in 2021. That obviously did not prevent him eventually from getting COVID-19, probably with Omicron. On May the 10th, he had it for a week or so. Okay, so he would have developed an immune response to, should be the spike protein initially, and then when he got infected with the virus, he should have been making antibodies against the spike protein again, right? Because it's like a second shot, a booster shot, except he's getting it naturally. And, he was probably exposed to the virus before that in between anyways, but he got, for a variety of reasons, the COVID-19, his resistance could have been down, he could have been sick, could have been stressed, could have not enough sleep, nutrition. There's all kinds of factors that can come into play. Maybe it's the first time he encountered Omicron, which seems unlikely by May 10th. But let's say he did. He should have had a good spike protein response. So then he got vaccinated again on July the 20th. That should have been like a 3rd shot, at least, to induce his spike antibody response. But when we look at this plot, what we see is a very good membrane protein response and against the other non-structural proteins in the virus. So clearly he had a good natural immune response against the other proteins that were in the virus, but his response to the spike protein is amongst the weakest that I normally see in people that have natural immunity, that have been infected with the virus. So I think this data, and I'm paying attention to this for more of our participants, I think this data is really pointing out to me, that if you are vaccinating after you already have a fairly robust immune response, remember when you're vaccinated, it's not like when you're normally encountering a small number of these viruses and you're getting a localized response that in fact is not a huge infection in terms of number of particles. When you're vaccinated, you have trillions with each vaccination of mRNAs that are producing, again, it could be 50 to 100 copies of the spike protein from each mRNA that successfully gets inside your cells. So it's a real robust effect. So what you're probably doing is you're turning on what we call tolerance mechanisms or antibody-dependent enhancement, where now you get sick right after you've been vaccinated, because you've actually made it easier with the antibodies that are present. They're binding to the virus, and then instead of the virus where the immune cells are eating the virus and digesting it, they actually are attaching to these viruses to the immune cells, and instead of going through their digestive system, they're actually entering into the cell and actually replicate and create more virus inside the cell that kills the immune cell. The very immune cell that's supposed to be producing the antibodies or providing protection against the virus. So those memory B cells we talked about and plasma cells that normally would kick into action when you re-encounter the virus and make more antibodies to protect you. Well, by doing this immune response to the vaccine, you may actually be turning on a program where they commit suicide, because they think, these cells, that this is part of the normal environment, that there's not a threat here. And so what ends up happening is you induce immune tolerance. This is the way that your body will normally recognize your own body proteins, so you don't make antibodies against yourself. It's the process involves the suicide of these antibody producing cells so that you don't have autoimmune disease. And so now your immune system is thinking, well, not thinking, but acting as if this is your own body proteins, these spike proteins. And so they actually get rid of the best B cells and T cells that would protect you from COVID-19. And so ultimately, if we continue on this course of continuous booster shots, it'll be more and more like you'll actually reduce your natural immunity, and you will be more susceptible to future infections of SARS-CoV-2. And that's why this strategy, it has to stop. It's actually causing more harm than good. It's actually increasing the likelihood that people that are vaccinated will actually be more susceptible to getting COVID-19 and its variants in the future. So I'll leave it at that, and I'd be happy to answer any questions that you may have.
Thank you so much, Dr. Pelech. I'm sure a lot of people have questions, and I'll let them definitely talk. But if everyone's okay, can I ask my questions? So initially it was being called the pandemic of the unvaccinated. Now,
Right,
is
yes.
it fair to say that now we are in the pandemic of the vaccinated?
Well, absolutely, sure. Absolutely. I mean, I think at the statistics now is probably at least 95% of the people that are in hospital that are dying are double or triple vaccinated. So to think that a small percentage, I think it's probably last I had seen, certainly in British Columbia, only 13% of the population was unvaccinated. So when you have a situation where there's such a small percentage of that population is unvaccinated, and most of them have natural immunity, so it's even less likely that they're going to get infected to the point where they're going to be contagious. I think you can conclude for sure that we are now in a pandemic of the vaccinated.
Right. So I think I know a lot of people want to know the obvious question since you're talking about Kinexus. Can you walk us through that process of what happens when people, and why should people choose to get their antibodies tested? And very briefly, what to expect when they send their samples to
Sure.
you? And also the third one, if I may ask, would you be open to giving letters to people if they are, showing that they have high antibodies and that they do not need to be taking these shots, especially students who want to continue their studies and the schools are forcing them to take the boosters or whatnot? So if you can speak a little bit about that, please.
Sure. Well, firstly, my general advice to people is that if you don't really need this information because of your job or travel or something like that, I wouldn't bother. I mean, you can assume that you have been infected and you have antibodies. If you've been vaccinated, then there might be some question, like we showed you in the case of this example. I mean, the problem for this person is, the good news is that he has membrane proteins. So he does have antibodies against the surface of the virus, and he'll have some protection from that, but it won't be from the spike protein. But not everybody makes antibodies against the membrane protein either. But for most people, I think it's safe to assume that you have been infected, that you have an antibody response, and that you're actually fairly protected at this stage. If you decide that you want to get tested, we have two options. One is that you can come to Kinexus if you're in the Vancouver area, and we can take a blood sample. It's a pricking your finger and taking a few drops of blood, and that's all we need. And then in about 2 to 3 weeks, we give you your report, much like you saw that immunoblood image. You'll get your own immunoblood image with the 41 marker test for looking whether you have antibodies against 10 of the SARS-CoV-2 proteins. So the other alternatives, if you live outside the Vancouver area, is that we send you a blood sampling kit. So basically the kit includes 2 lancets that are sterile, that you just push it, you just like place it against your finger, push a button, and it's a very tiny little pin that you'll get a couple of drops of blood that will come out of your fingertip that you you spot onto a protein saver card that you then mail back to us. And so the cost for the analysis that we do is $85 for the 41 markers, and the sampling kit is about $14, and the shipping the kit to you is about $2 by post in the mail. So that's the cost. The turnaround time is about 2 to 3 weeks. We're trying to keep it within that you'll get your results back. Now, in terms of a letter, anybody who's in our study, if they've registered, then I'm happy to provide them, if they ask for it, a confirmation of registration letter. And if we have your results, I incorporate those into the letter so that you can show, whether it's the university or your employer or for travel, that you are part of a clinical study looking at antibodies that's been ongoing by a company. It's previously had ethics board approval. It doesn't, right, for their second stage, we haven't bothered because we're adopting the same procedures. So what will happen is someone will see that. They'll see, because I'll give an example of what the blood looks like for control, that, yeah, you're just loaded with antibodies that recognize different parts of the virus. you're clearly got natural immunity that is going to be, you're less likely to get sick and you're less likely to transmit to somebody else. And it's actual evidence of immunity. That's a lot different from saying, oh, I've been vaccinated. Oh, that was 6 months ago. I might have antibodies, or I probably don't have antibodies. Who knows? But if you've actually showing, we're measuring the actual antibodies against that particular virus, then it's actually much more reassuring to your employer or the university to have that kind of information. Unfortunately, it's not really recognized natural immunity in Canada. It is, though, in many countries around the world. Yeah, and actually, what's interesting is now in like Sweden and Norway and United Kingdom and Germany, they're not even recommending vaccination of kids that are under even 18. And now Denmark has just announced they don't recommend vaccination for people under age 50, period. Unless they're very, very high comorbidities, they don't recommend it at all. They have to have like a doctor's note now to get that permission. So that's where we're at now. But Canada, let's start vaccinating the 6-month-old babies. Based on a clinical study where the difference between the placebo group and the vaccinated group is one single child. It's just insane. Okay, Sarah, I think you're muted, so we can go
Oh, well,
on.
I'm just saying that how do we get there? I think that calls for a whole new session.
Yeah.
But maybe after people have asked their questions, I do want you to touch on the point that these are really not vaccines, are they?
Well, we changed the definition of a vaccine. So the bar to be accepted as a vaccine has really been lowered. So a vaccine is any product that will stimulate the immune system to fight disease. They don't even say an infectious disease because, again, we are developing vaccines against cancer, so that's not infectious. So they've lowered the definition. And the problem is that, well, that definition applies equally to yogurt, as my colleague Byron Bridle will point out.
I'm having a vaccine.
Yeah, but the problem is that the liability for the manufacturer for a vaccine in the United States is much lower than an actual drug. And the RNA vaccines, the adenovirus vaccines for COVID-19, they are genetic vaccines. They are genetic therapy, but we've not applied the same standards that we would normally apply for genetic therapy products, which are much, much stricter than even for drugs. So this is the crazy situation that we're in, that we're now approving these vaccines for younger and younger children. We've got 6-month-olds now, which, is based on very small studies, very short duration, with very little direct measurement of potential harms by the way of, for example, biochemical tests, like for troponin T to see if there's damage in the hearts of these, young children and babies. Right. So we're making it so easy. These are called these kind of studies where you're called bridging studies. So if you have an age group where it's been tested more, and all you have to show is that, well, you're getting an antibody response as good as what you're seeing in that other higher age group, that's sufficient for its approval. And that's exactly what happened.
Yeah, we've really dumbed down science. But anyways, I'll let Bernard and then Carrie ask their questions, because I could just keep going on and on.
Hi, Stephen. Thank you very much for this excellent presentation. I'll try to refrain myself from asking too many questions, but I have a lot of questions. So let me start first by your initial work you've done in order to examine which to me should be the foundation of any vaccine program.
Right.
You're going to design a vaccine, you'd like to know what kind of immune response this particular pathogen is triggering.
Mhm.
And then you'd like, this is not done, but you'd like to correlate whatever you measure in people with protection, right?
Right.
Because right now it seems to me that we're playing really, really loose with this antibody titer as a correlate of protection, because we don't know that.
That's right. And actually it's interesting. I didn't go into the nuances, but there's so-called neutralizing antibodies. Those are the ones that
Yeah.
target just the receptor binding domain. And their assays now is not even looking at your overall antibody response against the SARS-CoV-2 virus, which can be very protective, but they're focusing in on just the so-called neutralizing antibodies, where they bind to that receptor binding domain and interfere with the ability in studies where they're not even looking at the virus. They're looking at
I know.
lentivirus
Yeah.
constructs that have
Yeah.
the spike
Yeah.
protein
Yeah.
and using that as a
That's
surrogate.
different. That's totally
Yeah.
different.
They're so far removed from, well,
Yeah.
does it actually prevent an infection of the virus in people?
So I guess I don't want to get too technical, but
Mhm.
it seems to me that the technology that you've developed is, yeah, it's probably cutting edge and so on, but
Mhm.
I wouldn't be surprised that similar technology of that nature would exist in other biotech company, or is it that unique?
Spot synthesis, where we make these artificial peptides.
Yeah,
that
yeah.
way. There's probably maybe, I would think maybe half a dozen labs in Canada that can do this, like academic labs. Industrial labs, I don't know of anybody else in certainly the lower mainland of British Columbia. But the technology has been around for about at least 15 years.
That's my point. My
Yeah.
point is that because the technology is there, I mean, how can you not do this kind of study if you want to design a vaccine? That's just maybe more of a comment than a question, but
Right.
to me,
No,
it's
you're right.
the other thing that I had, and it really, to some extent, this is a question about the dynamic of having infection, which we don't know unless we test it, right?
Mhm.
And when you test in the what they call the real world, when you deploy the vaccine, unless you test whether people have been infected or not, what kind of conclusion can you bring on whether the vaccine had any effect on protection if people were already protected?
Yeah, well, this is the central point is in most of the studies that have been done, even the clinical studies,
They
even
don't
with
know.
children, they did not evaluate whether or not they had natural immunity in the first place.
Yeah,
So you cannot tell whether or not the effect you're seeing is a difference in maybe the proportion of people with natural immunity that are different in your two populations. So this is a problem for both studies with drugs to look at the effect to reduce, if you get COVID-19, are these drugs effective? They're not looking at the natural immunity backdrop. And of course, for these vaccines, again, they're not, because this tests are so insensitive, even when they do apply it, they still don't actually know whether or not this individual already had natural immunity. And maybe the vaccine boosted it, but as we can see from the work I'm showing you, it looks like it's actually maybe be reducing it, right? So and it's a moving feast, right? Because as we have more and more people get infected and more variants, then unless our tests are very accurate for those newer variants and detecting our immune responses, we're not going to be able to tell. So for example, our test is based on the Wuhan strain. We have to make a whole new test maybe in the future for Omicron, B20. It might be so different that our test won't work. But so far, it's working pretty good.
So, okay, maybe one last question, because you focus all of your analysis on antibody response.
Right.
It seems to me, though, that the T cell response is probably playing a major role. in protection.
Absolutely. And actually,
And can we monitor that with some sort of TCR and assay that would give us some insight in terms of generating the right T cell?
right. Well, my colleague, Ismail Samide, he's the president of Immunity DX in the Vancouver area. And he developed T cell testing for SARS-CoV-2. And he's done, I'm sure, a few hundred people, but we gave him samples from people early in our study, blood samples, where we got the T cell population, peripheral blood mononucleocytes, and we gave it to him. So he went and looked at the T cell response in those T cells from the very people that we looked at the B cell response with antibodies. And we found that they correlate really well. If we had a good antibody response, he had a good T cell response. If he had a poor T cell response, we had a poor antibody response. So I think the antibody testing is a good correlate, or like a marker for what the T cell responses are like. Because as you know, Bernard, you do need some of your T cell responses to get your B cell responses going in the first place. So what ends up happening is, unfortunately for him, because the costs of his test is around $300 just to do the test, never mind the blood sampling cost, he's just not been able to be successful as a business. And he's actually is winding down that T cell testing. So I'm not sure where else you can do it in Canada. And I've heard, I think it was ICOR Blood Services, I've heard from multiple people now that they're no longer in business. So it's not that many places you can do even the commercial testing of the antibodies. But in our case, we're doing a clinical study. We're not offering this as a commercial test, but we are making it available to anybody who wants to be part of the study.
I want to be part.
Yeah. So, and this way, I think it also helps people that need a waiver and a reason. Usually, if you're part of a clinical study, that's respected, at least in academic institutions, that there is going to be the control population, and we need to have those controls. And there's very few of us that are left. Yeah.
So, Steve, have you had a lot of vaccinated people? I don't even want to call it vaccine, but a lot of people with the mRNA shots come for testing, or is it mostly?
Yeah, more so now.
Yeah.
Interesting. I'm seeing one of the groups that's starting to really, by word of mouth, is actually people in the movie industry and TV. Because when you have a person sick with COVID, if they're an actor or they play a critical role, it stops production, and it's very expensive, everyday delay. And so they've been very active at monitoring people that are unvaccinated with rapid antigen tests and the like, usually at their own cost, or now even insisting that their actors and other production people are vaccinated. And it's interesting because many of them do not want to be vaccinated. They've been doing their homework, and they don't want to put themselves at risk. And so they're trying to get into our study, in part so that they can go to their employer and say, look, I'm being monitored, I have antibodies, I'm part of a clinical study. And the funny thing is, I think at the end of the day, those actors and production staff that have decided not to get vaccinated are going to be much healthier than the people who decide to get vaccinated, which are going to be at higher risk of getting COVID-19 in the future and getting quite sick. It may actually work, like opposite to what was intended by the, seniors in the movie industry. And certainly they're pushing their unions to kind of like push back against management that's insisting on mandatory vaccination wherever possible. So yeah, it's an interesting situation that they're stuck in. And I think I just read this morning there's many industries where this is problematic. The airline industry is another. I think I just read a newspaper article this morning. One of the airplanes, the pilot died while he was flying the plane.
Many pilots have died in while flying planes. I was reading this thing with Free to Fly, and they said they've been pilots for like 20 years, and in their entire career, they've had like 2 pilots who have died, but in 1 year, they've seen, I don't know, 6, 7 of them die.
Yeah.
So
Well, this is a
crazy.
pilot today. It's
Yeah,
made the papers.
yeah. I'll let Carrie ask. She's been patient. Thank you, Carrie.
Hi, Carrie.
Hi, thanks for talking to us. I actually have 3 questions.
Mhm.
One question is, if someone is taking a preventative antiviral, for example, ivermectin,
Mhm.
will that change any part of the antibody response if they've been taking it consistently?
It's a very good question. Occasionally, I get blots with blood samples from people where I don't see much of an antibody response. And I thought that was very interesting. And then when I investigated, these people are often taking something like ivermectin prophylactically. So to prevent from being infected.
Right.
And so obviously that prevents them, maybe when you got a pandemic and it's really being spread around, that you might be concerned being at high risk. But the problem is you're not going to develop a good immune response if you don't give a chance for your immune system to really get to know this virus. And so my recommendation is that if you're very, very high risk, okay, I think, you can try to make a case that you might just have to take ivermectin for the rest of your life. But I think for most people, you're better to take the ivermectin if you want to do that, or whatever medication that you think is the best. Do it when you just start getting sick, or maybe you're like I said, at high risk, or everybody in your family is really sick with COVID-19. You might do it, and maybe not even a dose that prevents you from getting sick, but a dose so it doesn't get too bad before it happens. But I think in general, you're better to not take medications unless you have to, and I think it is important for your immune system to encounter the virus. The good news is pretty much everybody has been infected. I mean, it's very rare I find someone who hasn't, but when I do see it, sometimes it's just like you described. This person's been taking ivermectin regularly, which actually is a great endorsement for ivermectin. It really is doing something.
Yeah, that's great. Thank you. How many people did you do in your first antibody study, and how many have you done in your second, and what is your max?
So we've had 3,500 people roughly in the first study. And I think we're probably pretty close to 500 that we've done, recently. We can certainly take on more people. That's not a problem. We can ramp up. The big problem would be me, because I write the reports for everyone and I prepare the letters if they ask for a clinical study registration letter confirmation. And so I have a lot on my schedule, but I have great people that are working with me that work very hard. So at one point we were doing up to 80 samples a day. So we can do that if we have to, but right now it's a very, very manageable number, and we can certainly take more people. So I'm not putting a max at this point, because the more data we accumulate, the better the study actually is, and you can start to get to the more nuances of how many vaccine shots, how long does it last, which markers are the indicator of the different parts of the virus that might correlate best with the vaccine compared to with the natural immunity. And there is some slight differences that we're already seeing, but the biggest thing is I need more people who have had COVID-19 and then got vaccinated. This is what I'm really concerned about, because let's face it, pretty much everybody's already has natural immunity. So now if we're going at it continually with these booster shots, we may actually be destroying their immune systems.
Right,
Mhm.
right. Okay, and my last question was, you said that you didn't receive a grant for your study.
No, I applied.
So how does it work in Canada when you're doing a study? Like, are you registered somewhere in Canada? Like, how does that work?
Well, it depends on the kind of study you're doing. If you're looking at a drug, or you're maybe a vaccine, if it's something that's invasive into the person's body, then you have to get approvals, and it has to be an ethics approval that includes looking at what are the health consequences, risks for those individuals that are your participants. So we did do, in our first study, we did get ethic board approval through Veritas IRB in Quebec. But in order for me to publish my results, because the kind of study we're doing, we're not going into a person's body, but there are ethical implications of, where confidentiality is very important. So we maintain all of that. So when we want to publish in a really good, reputable journal, then it is important for those studies to actually have ethics board approval. However, you can still publish results. So in our first study, we'll be able to do that. In our second study, we're probably just going to go ahead and publish it as a white paper, or we'll publish it in a journal where they're not so concerned about that. So it just depends. But right now, there's only 3 people in our company that match the data to the person, and the results are confidential. I can give you a letter with your results. What you do with that letter is your business. And, it's got my signature on it that I can verify the information that's in that letter is from me. But, if you want to keep it private, you keep it private. If you want to show it to your employer, you can show it to your employer. There's no restrictions in that regard.
Right. And have you had contact from people, I guess, around the globe who've just been interested in what you're doing, like other scientists?
Well, we've published some of our results in collaboration with the BC Women's and Children's Hospital. We showed with about 276 healthy adults in May of 2020 that 90% of them had antibodies against SARS-CoV-2 spike or nucleocapsid protein. So that publication has actually started to be fairly well cited. But the implication at the time was we thought maybe it was cross-reactivity with earlier coronaviruses that these people had that happened to recognize SARS-CoV-2. But I'm convinced now, having looked at more than 10 times that number, these people actually had SARS-CoV-2. Yeah. And half of them were healthcare workers. So it's not surprising, right? I mean, in a hospital setting is where you're going to likely get your highest exposures to this virus.
Yeah. Thanks so much.
Thank you for the question.
Bernard, I'll let you ask, but before that, Hannah's asked me to ask that question on her behalf. For some reason, she's not able to raise her hand. So can you, she's asking, you, Steve, if you got Bonnie Henry's job, knowing what you know, what would be the first things you would do to improve the situation? And what recommendations do you have for the government to do?
That's a long list. So I'm not going to go into that, but I can tell you that the first thing we've got to do is do a real audit of the effectiveness of the policies that were already done, the masking, the
distancing,
quarantining, the social distancing, shutting the borders, the vaccination programs. All of this needs to be really, there's so much data now that's actually out there, and they have it. So, we need to find out what were our mistakes, and then we need to use that information to really guide us in the future, and also pay attention to the data we had before, like for the influenza, for example. It was never recommended that we have masking in a pandemic with influenza because there's such a wealth of data from decades
Yeah.
showing it doesn't work.
Yeah.
So, so it's one of those things where there's a lot of work that has to be done. And also one of the things that I would do is I would separate the health regulatory agencies from funding from the manufacturers of these products so that they're completely arm's length. And anybody who's been in that industry, they can go into a regulatory agency, but they can't come back out and take a job now in industry again.
Good
If
one.
they decide to go down into, and that's very difficult to control because you can't deny someone, their livelihood. But I think you can, many companies have a non-compete thing that if you're an employee of the company, you can't go out later
Yeah, to the
to your
competitor.
competitor.
Yeah, yeah, yeah. No, the
So they should
airlines
have those
industry has
non-compete
that.
agreements.
It's there. It's happening in other industries. So, and I think and I'll let Carrie Bernard. Is that okay if I let Carrie Agape ask her question before you, just because she has not asked one, and then we'll get to Bernard, and then later on, just before I forget, if we can just quickly touch on why did the government not do antibody testing, right? They kept doing PCR. So Carrie, go ahead.
Okay, can you hear me?
Yes.
Perfect. Okay. Yeah, I'm just wondering, it seems like at some point in time I'd heard that the antibodies to the spike might also themselves do harm in the body. I was wondering if you'd heard that. And yeah, I was just
Yeah. So yeah, the concern is that if you have a very narrow focus of the antibodies that you have, they may not be quite as effective at taking out the virus as a multi-pronged approach with different antibodies sticking to different proteins in the virus. So your immune response is very targeted. And with that very targeted response being not quite as good, you have the danger of the antibodies sticking to the virus. And as I was mentioning earlier, may actually facilitate the entry of immune cells. Well, so the virus, the antibody I showed you, it has like a shape where you have these outside parts and then you have a central part. That central part is similar in all of the different antibodies. The part here, we call it variable regions. They can bind to, they have affinity for, let's say you have two viruses. One antibody can stick to two viruses, actually bring them together if sterically they're small enough. But this part here, the base, that all of your many of your innate immune cells, macrophages, other types of phagocytic, immune cells that eat other foreign viruses, bacteria, they have receptors that actually bind that part. They're called FC receptors. And those FC receptors are on the surface of some of these cells. So instead of the virus sticking to ACE2 and once latching in, getting inside the host cell to replicate, it can actually get inside of the immune cell that was actually going out to try to attack it in the first place. And if it gets inside and replicates, it kills that cell. So that's one concern. The other is that when you have antibodies and there are very high levels of a particular antibody, it may target more than just the virus protein. It may actually start to what we call cross-react with other normal proteins found on the surface of cells in your body. And in doing so, tag those cells for the immune system to attack it. That's why this whole concept of these RNA vaccines and adenovirus vaccines, where you're putting your virus protein on the surface of your own cells, you're triggering an immune response. So not only are you potentially inducing an immune response against the spike protein, but against your own body proteins. But the hope is that you've developed tolerance so you don't have antibody producing cells that recognize your own proteins, because you've eliminated them from the repertoire of all these B cells that you are born with. So the problem is, and this is why antibody levels decline after the threat is gone. Because if you maintain very, very high levels of all of your antibody producing cells are all pumping out their antibodies all at the same time when it's not necessary, your blood would be so thick, it wouldn't flow properly, and you'd have like illness from that itself. And at very high concentrations, antibodies start binding to other things in a less specific way, and then you'll get a lot of autoimmune disease. So this is the balance. You want your innate immune system, your autoimmune system, your adaptive immune system with T and B cells to be turned on when you need them, and then turn them right off afterwards. But if you have very high levels of these antibodies, then you get off-target effects, and again, autoimmune disease. And in fact, this is one of the problems they're having with some of the treatments with antibodies that are made monoclonals against the spike protein. You know, someone gets sick and then they're going to infuse them with antibodies against the spike protein. Well, that actually seems to induce, again, this antibody-dependent enhancement. And so some of these monoclonals no longer work very well, especially if they're targeting the receptor binding domain and you have new mutants. But actually, they actually can reduce your immunity, because again, you're getting rid of some of your, what we call innate immune cells that are non-specific, but will recognize protein or viruses, bacteria that have a protein that antibody happens to recognize. So anyways, that's why some of these treatments and even convalescent serum sometimes doesn't work that well for treatment.
Yeah, Bernard, go ahead with your question, and I'm sure Steve is now getting anxious to get back to his family, but we'll.
Yeah, well, you mentioned that the vaccine waned pretty well after a couple of months.
Right.
But if somebody's been vaccinated, get reinfected, shouldn't that trigger the memory response without booster?
Yeah, I mean, this is what you expect. I mean,
Yeah.
why isn't the natural immune response, we know we have immune memory, and we've shown it ourselves in people like 2.5 years later. And certainly I showed you in those people almost a year later that they had just as robust a response. Again, I think it's because everybody is being exposed to the virus continually
More
in the
than
pandemic.
once. Yeah.
And so as long as that's happening, then these antibody levels remain high. But for some reason, in the vaccinated people, it'll decline. And so to me, that would indicate you're probably not establishing that immune memory, probably because those T and B cells are undergoing an apoptotic process. I don't know. But it just doesn't seem to work as well.
Well, you know the word that shows that there's a potential with the mRNA vaccine that the interferon response is temporarily downregulated.
Mm-hmm.
And the interferon is thought to be involved in the adaptive response as well. Could that be an explanation?
Yeah, I mean, there's a few different explanations been offered. Like I said, originally the antibody-dependent enhancement, original antigenic sin, the mechanisms of tolerance that we would normally develop, that your body can, for example, when you have an allergen, the best way to get rid of that is to give the allergen, in low doses but regularly. So the body thinks, oh, this is okay. This is not a threat. It's always there.
Class switch with IgG4, right?
Yeah, yes, class switch, exactly.
Yeah.
The IgG4.
But in case of the vaccine, is it possible that for some obscure reason, the abundance of the T cell, memory T cell that are important as a helper for the B cell response, as well as maybe potentially cytotoxic T cell, is not at the same level that it would be from natural infection?
Yeah, well, this is the thing. I mean, I don't know what the explanation is. It could be a combination of things,
Mhm.
but what we do know is there's no doubt that when you're vaccinated, you lose that immunity sometimes as fast as a month after you've been vaccinated. And typically around 5 to 6 months, at least after the second shot, but after the third shot, that duration seems to be shorter. So this is what they're seeing in Israel. That's why they had to go to the third boost, like the third shot, the fourth shot soon after they already had given the third shot.
So
But-
Steve, is it, I mean, we're only talking about the mRNA, but there's like the whole slew of that whole shot, that's poisonous too, correct?
Well, you can have an immune response to the lipids that are actually on the nanoparticles, but I don't think that's where most of the immune response that you're getting is from. In the case of these Medicago and the
Novavax.
Novavax, exactly. Here, you've got foreign proteins. They're actually on the surface of those exosomes, those lipid nanoparticles, and you will get an immune response to those too. Now, what will happen is the next time you get vaccinated, you will have probably more antibodies that are actually sticking to those particles. It might take them out faster, but you're at least bringing those immune cells to those lipid nanoparticles, which will include the spike protein. So in that case, it might actually be a good thing that you're getting your immune system able to recognize the vaccine early and the immunogens that are in it, and get a nice boost of your immune response. Also, you know exactly how much spike protein is in those, because you can quantify that. We can't quantify it with these RNA, well, these genetic vaccines. The difference between one person and another person on the levels of the spike protein that's produced could be 2 orders of magnitude, because it depends on which cells were infected, depends on the batch of the vaccine, depends on whether when you inject it, did half of it go into your bloodstream as opposed to the site of the injection into the muscle, depends on the cell type that the lipid nanoparticles got into, what's your metabolic state, what's your nutritional state,
Activity state.
all these factors come into play. Yeah, and at the end of the day, and genetics. And so at the end of the day, you may in fact have a 100-fold difference. So some people, they get vaccinated and they don't get any protection. They also don't have any vaccine injury.
So we
That's
think.
good news.
No, I was actually asking, we've been hearing about the graphene oxide and the EDTA in it and all of those, like the slew, the whole cocktail which carries this mRNA. I mean, is that not playing a role?
In your shampoo products and
like we're injecting
stuff.
shampoo in ourselves and nothing will happen.
Well, the dose of the vaccine is relatively small for that, and I don't even know there really is graphene oxide. I mean, this is what people have talked about, but I haven't seen really good data that mass spectrometry data. I would imagine that the amount that you're actually getting is going to be tiny compared to what people are typically getting in products they take every day, including like in ice cream, people were getting all upset about polysorbate 80. I mean, that's one of your major ingredients in ice cream.
So anyways, we're going to start to end it, but I don't want to leave with this impression that people, or is it true? Like should people be thinking that Medicago and Novavax are kind of fairly safer than the rest?
Well, no, unfortunately, I believe there's data now for cardiovascular disease. I think, I can't remember if it was myocarditis or thrombosis, but actually the risk is actually very similar to what you have with the genetic vaccines. And so the implication is that the actual production of the spike protein itself
Mhm.
is problematic.
Yeah.
Mhm.
And that may be engaging platelets and other types of cells to affect, the really platelet aggregation, clotting. So, one of the things that's really fascinating is that when these vaccines are made, they were mutated, the spike protein, to have these extra proline residues put in it to lock it into a pre-fusion state. But they left in the furanose protease cleavage site so that you can cut, on the surface of certain types of cells, like, liver cells have a very high level of this, that actually make it releases the spike protein so that you can still have part of it engaging receptors. Even if you have normally the vaccines produced where the protein's kind of like anchored to the surface of the cell, it shouldn't be floating off into the bloodstream. But if you got, cells that have the furanose on them, then they can cut them. And that could allow actually the release of a portion of the spike protein that might still engage receptors, for example, and activate them. You don't have to get inside the cell. You just have to stick to the receptor to activate the receptor. And there are many other receptors for the spike protein besides ACE2.
Mhm.
Neuropilin is an example in the brain, but there's others. So, and there may be more that we have not yet identified.
So when you get the virus, I mean, wouldn't people argue, so if you get SARS-CoV-2 naturally, then you would still make the spike protein, right?
Well, it's in the virus.
Yeah.
And yes, you'll have for a short while the virus is assembling the particles on the surface of the cell, because when you make a new virus particle, it'll take the proteins and the RNA that's necessary in the virus, but it'll all move to the surface of the cell, and then it kind of like bleb out, taking part of the cell membrane with it. And so while it's there on the surface, yes, your immune system will recognize and take it out. That's where your T cells are particularly effective. For cells that are actually have the foreign proteins of the virus or bacteria on the surface, then the T cell kills those cells to prevent further production of the bacteria or the virus in that cell.
Right. So
So-
you wouldn't have these antibodies continuing to circulate or in the body through a natural infection, because your antibody
Well, the
levels.
antibodies are okay. It's
Yeah.
just, are you producing more of the spike protein in
Yeah.
a form that it can travel easily and
Okay.
evade the immune system? But again, there's stability issues with these proteins, and there's natural proteases that like proteins that clip. And in fact, when you're talking about a gastrointestinal tract infection, you got all these digestive enzymes that are actually in your gut that are like, destroying all these proteins in your gut.
Mhm. All right. Well, this has been absolutely amazing. Thank you so much. We've run over time. I totally expected it. So thanks everyone for signing in. And next week, let's come on and listen to Joseph Borgo from Canadians for Truth and learn more about natural methods to treat ourselves, just like Dr. Pelech mentioned, that always using natural methods is much better than taking medications, correct?
Yeah, no, I think medications that are drugs, they're normally well tested, and they're used as appropriate for the circumstance. But I think, it's a good idea to have good nutrition, good exercise, good sleep, which I could do with. And also, there are a lot of medicinal ingredients that are actually naturally in products that are very protective. And so it's kind of interesting. People take supplements like vitamins. They think, okay, these vitamins, I don't need to take my fruits and vegetables and nuts. I got all my vitamins in a pill. And then I just have, the rest of my diet that's, actually could be high in carbohydrates and fats. But what people have to understand is that there's over 600 micronutrients
Mhm.
that are found in fruits and vegetables and nuts that actually provide essential ingredients for a healthy body, but you don't need very high doses of it. So they're in a sense like vitamins,
Mhm.
and you don't get them when you take a vitamin supplement. But if that's the source of all of your these nutrients that you need, then you're going to be deficient.
Yeah.
So
And everybody needs different ones, and that's where you know we start to, when they start talking about personalized medicine, now it's mostly about personalized drugs. But I think personalized medicine is, well, would tomato work for me, and would red chili pepper work for me or not, or and we
Yeah.
figure it out, right? So for everyone who wants to know where the recording will be, I've posted the link to the BitChute channel in the chat, and I will do my best to put this lovely talk ASAP. And there are many other talks from our previous sessions over there. I'm doing my best to have everything because YouTube shut me down. So all my previous talks are gone. So, well, we're working on it. Right. So thanks, everybody. Thanks a lot, Steve. And
thank you, Sarah.